The global landscape of invasive mycology is fracturing. Advanced immunosuppression and broad-spectrum antibiotics allow rare yeasts to multiply and thrive in vulnerable patients. For decades, Candida albicans was the primary focus of clinical mycology. Now, its relative prevalence is shrinking. Fungal genera like Trichosporon, Rhodotorula, and Saccharomyces are expanding into that space. To profile this shift, we aggregated the latest clinical guidelines and mycological literature using a Gemini deep search, compiling the source data in a Notebook LM document. When diagnostic tools designed for common Candida encounter these rare yeasts, the identification often fails or returns a generic result. This creates a gap where an infection is present, but its specific biological identity remains hidden. Managing these pathogens requires navigating a historical hurdle. For much of the 20th century, mycology used a dual nomenclature system that assigned two separate names to a single organism. The anamorph branch describes the asexual, budding state, while teleomorph describes the spore-producing sexual stage. Under the one fungus, one name principle, these merge into a single genomic identity. Yet yeasts like Trichosporon asahi remain caught in transition, existing strictly as anamorphs. This is distinct from an organism like Saccharomyces cerevisiae. It is recognized as a complete holomorph, meaning its asexual budding stage and sexual ascospore stage have been identified and mapped under one name. Precise fungal nomenclature determines the patient's treatment path. Using an obsolete name in a lab report creates a susceptibility assumption that doesn't match the organism's biology, resulting in the selection of ineffective antifungals. This matrix compares Candida albicans against emerging rare yeasts. Candida relies on high primary virulence, actively penetrating host tissue. Conversely, rare yeasts lack these tools, acting as low virulence opportunists that rely on external breaks in the host's defense. They enter the body through passive translocation. This occurs when anatomical barriers are breached by medical interventions, specifically through contaminated central venous catheters, or when a compromised gut barrier allows the yeast to slip into the vascular system. Once inside, these yeasts deploy defensive adaptations. Trichosporon asahi constructs structural biofilms, while Rhodotorula uses a protective capsule to evade the host's immune cells. While these pathogens lack the invasive machinery of Candida, they cause high mortality disease by exploiting the patient's own medical support systems. The population at risk is well defined: patients with profound immunosuppression, those receiving total parenteral nutrition, and extended-stay ICU patients. Rare yeast fungemias represent a small fraction of fungal infections globally, but their incidence is rising within these specific nosocomial settings. Consider Saccharomyces boulardii, a yeast widely administered as a protective gastrointestinal probiotic. Administering this probiotic in an ICU can trigger outbreaks. The yeast can spread from the probiotic capsules to the hands of healthcare workers, eventually contaminating the central lines of patients who were never prescribed the treatment. In this context, the modern hospital environment acts as both the primary incubator and the vector for these infections. Standard microscopy and India ink preparations are far too nonspecific to differentiate these organisms, creating a critical diagnostic bottleneck. Clinical laboratories now rely on MALDI-TOF mass spectrometry. By analyzing the protein fingerprint, it identifies a species within minutes.
This illustrates the MALDI-TOF matching process. Accuracy is restricted by the reference library. If incomplete, rare yeasts remain unidentified. Laboratories must constantly expand these databases with new spectra to maintain accuracy. When MALDI-TOF results have low confidence scores, labs must use molecular sequencing. Targeting the ITS and D1-D2 regions provides definitive genetic identification, though it significantly increases the turnaround time. Clinicians must also watch for serological traps. The presence of Rhodotorula mucilaginosa can trigger a false positive on the Aspergillus galactomannan assay, potentially leading to incorrect therapy. New methods like MALDI-TOF nucleic acid mass spectrometry are being developed to detect yeast DNA directly from samples, bypassing the culture phase. In mycology, misidentification misdirects the clinical response, often leading to the use of ineffective antifungal classes. These emerging pathogens are assigned by their multi-drug resistance profiles. Trichosporon asahi carries an intrinsic resistance to echinocandins, while Rhodotorula species frequently resist both echinocandins and fluconazole. This resistance is encoded in the cell. Point mutations in the FKS gene alter the target enzymes for echinocandins, while changes in the ERG11 gene prevent azoles from binding to their targets. Because these species are rare, there are no official clinical breakpoints to help doctors categorize a strain as susceptible or resistant. This scatter plot shows the distribution of minimum inhibitory concentration values for a rare yeast population. Without official breakpoints, labs use epidemiological cutoff values, or ECOFs, to separate normal wild-type populations from resistant mutants. This resistance landscape forces specialists to move away from standard empirical therapies. Using fluconazole or caspofungin against these organisms often results in clinical failure. Upon detecting a rare yeast, the first priority is not a drug, but aggressive source control. This requires the immediate removal of all central venous catheters and the cessation of probiotic therapies to remove the physical vector of the infection. The Dutch SWAB guidelines provide a framework for managing these infections. They emphasize the need for a coordinated clinical and pharmacological response. The guidelines recommend initiating early empirical therapy based on local epidemiology while waiting for definitive susceptibility results. Because of high azole and echinocandin resistance, lipid formulation amphotericin B is frequently the backbone of treatment for these Basidiomycetous yeasts. Survival depends on executing both tasks at once: rapid mechanical source control paired with aggressive, non-standard antifungal therapy. Mutations rendering standard drugs obsolete have created an urgent need for novel antifungal pipelines. Ibrixafungerb targets glucan synthase at a distinct site, bypassing common echinocandin resistance mutations. Similarly, fosmanogepix inhibits the GWT1 enzyme. This destabilizes the fungal cell wall assembly, causing the cell to lose its structural integrity.